Good afternoon, everyone. I'm Henry Tan. I'm an artist in residence right now. Today I will talk about the topic of the pursuit to understand life and bioaesthetics through an artistic research and creation part 1.5. Because when I start the residencies in last years, I gave a talk, the same talk, but it's part 1. So this time I have to make an update. And these are my names, known by different places, and I'm officially from an artist in residence at NYUAD, because I'm on the website. I just want to make sure that I'm legit. Okay, before I start, I would like to chant this memorial chants. A, T, G, C, A, T, G, C, A, T, G, C. A, T, G, C, the alphabet of life, a code that shapes each creator strive. For science quest, they give their all, a sacrifice that answers the call. Let us honor their selfless deed, a tribute to their noble creed. For in their sacrifice, we find a path to knowledge, humankind. So this is the poem that I wrote for tributing to all the cells, artificial cells, life that we use for experiment, and we pay respect back to them. Today I will share what I'm doing, who am I, what am I thinking, what is he thinking, what did he do, what he's doing at CGSB, and then what is planned. This is very brief. If you want to take home with these messages, this is the message to take home. But if you don't want to take home, it's fine, totally fine. And then the last message is he worth a contract extensions or another fellowships? So it's not about me, it's about people that I work with, and then the community that I'm from. Start from acknowledgement with the Freak Lab communities in Thailand, Subsunton Lab in Thailand, and also Metaforest in Japan, and then also my teams at Henry and Partners, and also we're here down here, CGSB teams, and everyone here who are supporting all the knowledge sharings and all the times that are very precious that you guys shared with me. I want to share one of these inspiring piece that kind of influenced my works. This is one of the statues, which is a statue for artificial cells and life by Professor Hideo Iwasaki. And then in these stones, he inscribed different symbols of life, which is DNA helix, lipid bilayers, ribosome, and eyes. And then he also inscribed that he acknowledged that these stones would help us kind of understand and rethink of how we understand the meaning of life, to understand the conditions that we are made to experience vitalities, as well as the history of the idea of life. So in part 1, I present this kind of my own pathway of understanding and try my pathway of research and explorations to art and science and to understand everything. But this is my overall map and pathway at CGSB. It's very complicated. This is, I think this is the same way that I feel when I look at your guys' diagrams. So I kind of narrowed down a little bit, but still confusing. So my main concern is about life, and then the intersubjectivity of life, try to understand what life means, and then from materialist view through panpsychism view or more animistic view, how do we approach to understand the meaning and the knowledge of life. That's why I'm not only looking through the histories, but also have to look at the scientific perspective as well. And one of the key concerns is about memories. I began to curious about what is the formations of the memories, what is the form. As an artist, we try to understand the form. What is the form of memories inside our brains? What is the mechanism of how memory is formed? And then I also kind of very much influenced by this aging society's problem that happened in Asia, and also myself, how we can address these problems to my research. And then one of the key what is the key? Anyway, I try to understand how life adapt and change into different environments, and then that causing these aging societies or memory loss or sleep deprived. So I kind of expanding into from sleep, I try to understand consciousness. From sleep, I try to understand circadian rhythms. From aging society, I try to understand, is it possible to technically reverse aging or aging mechanisms? And then with conversation with many scientists, and then they also suggest me many ideas, many possibilities, many theories. And then I always questioning these informations that come to me. It's also how I look at social medias, and all these informations also come towards me about supplements, new discoveries of drugs, new technologies of cure. So part of the project also expanded into the projects called Elixir of Life, that we try to understand how people search for cure in different time in the histories. And then perhaps at this time, the tools, it's in your hands, stem cell technologies, biotechnologies, and then how general public would look and perceive this discovery of these technologies. Is it the same way that how people just believe in animisms? And this is one of the questions that I try to kind of explore and link through my research and creations. Wait a second. So one of the projects that I've been creating last year is called the Pillar of Creations. Pillar of Creation is inspired from the Brahman, the Hindu god of creations. In Brahman's hands, he holds different tools of creations. One of the hands, he's holding the bowl, which is down here, which represents the cosmic energy of creations. Another hand, he's holding the scripts, which is represent the knowledge of creations. Another hand holding the rosaries, which represent the time of creations. And then lotus or spoon, which is represent idea of rebirth or sacrifice. He also has four faces, which represent different stage of man's life. So in Thailand, at these Enron shrines, people would go to the shrines to ask for fortunes, to ask for success. At the time that we with the societies that we are leans toward logical, evidence-based knowledge, and then how people still going to these shrines and then ask for monetary returns. Then I'm start to kind of try to investigate further on this process and mechanism and all kind of transactions that emerge from these shrines. Then I try to create these sculptures. This is not 3D models. This is actual sculptures. I try to reinterpret the tools of creations. Try to question back, like, if people really understand what tools that this god is holding, and then what does it mean? Or do you really understand the meaning of this god? Or you just go there because someone told you that they got a new jobs, or they won the lotteries, or they got new contracts. It's the same way that how we try to explore how this belief is emerged. With the cosmic bowl, the bowl that represents the cosmic energies, I try to create these brain organoids incubators. why brain organoids incubators? Because I'm very interested in the stem cell technologies, the possibilities that seems endless, the promise of new hope. But is it the real hope? Is it or it's just an advertising scam? So that's why I have to try to learn and understand the science behind stem cell technologies myself. And with another hand, he's holding the tools of knowledge. So I try to create these DNA printers. So it actually can print water, but not can be DNA. If you believe so, it will be able to print DNA. If you believe that it can, is it a DNA inside? It's a DNA inside there. Yeah. Another hand, he's holding the rosary bead, which represent the idea of time of creations. So I kind of inspired by this dementia clock, and so I create these 2 billion dementia clocks. The 2 billion system mechanism is try to keep itself as precise as possible, because it's kind of try to counter the gravitational force that happen to the clock. But the clock itself is a dementia clock. The clock itself forgets its own times. Dementia clock is the test why you are going for screening for the dementias or memory loss. People will have to go through like this drawing of the clock face, and then if you cannot draw like the whole face of the clock, like we see in a normal clock, if it's skewed toward one side, that means you have a sign of dementia. So what if the clock forgets its own time? What does it mean? And then in the process of the transactions, once people receive what they wish for, people will often go back to the shrines and then offer drinks and elephants or dance. So people would go there, offer very big elephant sculptures, or people will offer dance and performance. So I create these elephant sculptures as a way to question further what does these elephants mean, and then what does it symbolize. On the last weekend on the open campus day, there's one lady come to ask me, like, what is this? What am I putting inside this one? And then I told her it's DMEM F12 Glutamax. Of course, if you're not scientists, you have no idea what is DMEM. And then how much she asked, I said, it's $1,000 because it's the same price of the mediums. And then I told her that, of course, if you drink this, then you will kind of, okay, of course, you'll look young, you will rejuvenate, and then she really wanted to try it, but she doesn't want to buy it. And then she asked me if I take a Falcon's money. So no. So this project, Elixir of Life, as an elephant, is kind of try to question the idea of elixir of life, how people look at medicines, look at supplements, look at things that they consume, and then how we try to make sense of that. Like I would say my parents also heard about stem cell technologies or some injections that their friends had at a hospital, which cost very expensive. And then they, of course, after they heard that, they would like to try themselves. What does it mean? Is it real? Or is it the same way that the spirit doctors would just sell anything to anyone? How is this mechanism happen? So I'm trying to understand that. And also questioning myself after I learned about these professors that talk about reverse aging technologies, the promise of reverse aging technologies that he can make it in mice. He can use sirtuin or mTOR to reverse aging in mice with partial reprogramming. What does it mean, partial reprogramming? What does it mean that he can actually reverse aging? But then he also talked about he consumed this resveratrol, he consumed this NMN or NAD, and then I bought it. And then I have to try it myself. I have no idea how it works, and then I have no idea if it really works or not, but I already paid. Twice. Yes, even worse. So, where now? 10 minutes left. Okay. Going back to this one. So I've been trying to understand different things, the belief of people, how it emerged, how do we look at biotechnologies, how do we look at the cure, which is also kind of touched into what different labs at CGSB has been doing. So now is the part that I was showing what I've been doing at CGSB. Wait. I've been visiting labs, different labs at not only for CGSB. I also visit tissue culture lab, visit applied interactive media labs, visit astroparticle lab, visit artist studios, visit MFA, visit engineer lab, visit robotic labs. I try to know what people are doing here, try to see, I mean, it's more like the more I eat, the more I have ingredients for myself to cook, and then metabolites. Yes. I've been joining seminars, many seminars, and also a shout out to the team of Seminar Foods. I've been joining lab meetings. To be able to join the lab meeting is very precious time of my research and understanding to be able to follow how scientists actually works, solve problems, and how we can see how creative they are, see how they solve problems, how they struggle with the research, and then I have to also become part of that research to be able to really understand, truly understand, because if I want to create works that inspired from science, I don't want scientists to think that, oh, okay, this guy's come up with a crazy concept, but his technical understanding is really poor. So I have to catch up with the knowledge of these different research groups, which is really still really far behind. Then I met with this group called NYUAD. It's neuroscience clubs, and then we start to plan a series of workshops together, which I will share with you guys. And then I also get the chance to do a fire escape test, learn how to screen a zebrafish. I also try to ask, are you a wild type, or knockout, or mutant? What I got is a scratch. So science is hard, but cat is harder. And then I it's very exciting times to enter the lab, because it's very difficult to access the laboratory in Thailand. Some of the lab that I want to work with, the door is completely closed, but at CGSB it's really open. I mean, not fully open, but I can walk inside. But of course, now I still have to pass through all the trainings and also convince different groups, PI, that I'm qualified, I can handle pipette properly, I won't contaminate the experiments. But I'm getting there. Where? Yes. So from different activities that I had throughout these past months, there's one project that kind of inspired me. After I talked to this Arab Center for the Study of Art, they built this digital archive of Arab artists, and then after the conversations with them, we always discuss about the fragilities of works and memories, how they decide which artwork they want to keep or digitize, and then how many copies that they have to keep for the digital archive. And then they also have fear and anxieties of losing these archive as well. So I can feel that because I also have different copies of my digital informations. Then with the new technologies, the transfer of new technologies of these data always has some risk of loss. So these anxieties are always there. So I proposed them that, is it possible that we can store all the Arab art digitals archive in the living zebrafish? Perhaps together with UHRF1 and SS3MT and plus NM1 as well. And then see if it's proliferates together with the cancers, or if it's, what if the zebrafish reproduce, how many information can we retrieve back from, or maybe what if we release it in the sea or in the water, what would happen? Are we gonna get arrested? And then what if we put them into different gravitational force to speed up their life, or maybe slow down their life? Then what would it be? What does it mean if the people who are taking care of the archive also have to taking care of the fish? Because it's an archive of their life works, and I need budget for that. Then, of course, another project that I'm interested in is also try to learn how to grow and organize different organoids. Also try to create a system, the array to read out the signals from the organoids. So the organoids kind of conceptually represent the surveillance technologies that monitoring us. What if we can feed them with our anxieties? Would it differentiate itself into cancer cells, or it could reverse itself to become iPSC cells? No idea. But this is another project that I'm trying to do as well, which is cost more than the previous one. So along with these two projects, I also initiate this series of workshops, which is one workshop called GMO Omakase, GMOs. And omakase is like we cook what we have to serve you guys. So one of the workshops that I'm interested in is how to think about the epigenetic trace of arsenics or toxins that are exposed to our ancestor. Can we see these traces in our bodies? So this ancestor trauma is your epigenetic karma. In some cultures, they believe that what's happened to you is because of your karma. But perhaps we can look at your genes. And another project also called Cooking Memory DNA Storage. Also thinking about how can we store information into a DNA, can we cook them, and then can we revive them by eating this meat. And then another Dream Machine series. Next week I will have a Dream Visualize workshops with NYUAD and my colleagues here. We also will have a workshops on maybe talks about organoid intelligence, the rise of discussions on the consciousness of organoids. And then also another very intense Chronos drift, try to have an experiment of 5 days completely in the dark to see how circadian rhythm shift in the bodies. Oh, wait. For the hard organoids, actually I have a name for it, and then I already told Samira that if we do the hard organoids, that we can actually see its beating. It would be really interesting to see how someone has their own heart cells and beating outside the bodies, and then this project will name My Heart Will Go On. And another curiosity is I'm interested in bioelectricities, how another scientist, they also look at how different creatures have different charge of electricities that kind of governing how molecular expressions. Still have no idea. But anyway, after talking to Khaled, I was really inspired by kind of conversations with him. And then he said, I asked him like how different neurons kind of project itself, try to catch and build together and with another neurons. So you can see these small lines. And then the secret is in this complex system that have appear in all the beings, from the subatomic complexities to the cellulars, to organism, to societies, planetaries, and universe. So this is how I also inspired by the conversation with Khaled. I mean, if you look closer, perhaps you see nothing. But maybe, I see nothing, but maybe some scientists might see something. But if you look at the bigger view, maybe you might see something or you see nothing. Then, do I have enough time? No. Can I have 2 more minutes? No? Yes? Thank you. So perhaps I will end this talk with these videos that for the projects that we did last year.
My name is Henry Tan, an artist from Thailand. I'm one of the creators of the work Pillar of Creations, which is part of Emergence, curated by Fathomers. This exhibition is talking about art and synthetic biologies, and how synthetic biologies could give us a new definition of life. And today we have invited Mark Sogens from XIII to perform a memorial service to the living and dead organism inside the exhibitions.
Thank you.